Welcome, everyone. My name is Sarah Meyer, and I am a member of the HEAL Data Stewardship Group. Thank you very much for joining us today for the 21st installment in our Fresh Fair webinar series, A Thoughtful Approach to Open Science, Sharing Sensitive HEAL Data. Next slide, please. This webinar will feature a presentation introducing unique and less commonly addressed sensitive data types in HEAL research, followed by lightning talks from HEAL-funded investigators presenting how they have considered and made decisions around sharing results when working with sensitive data. The webinar will conclude with a live Q&A session. This webinar is being recorded and will be shared with all registrants. If at any point you have a question, please feel free to type it in the Q&A box at the bottom of your screen, and we'll bring it up during the Q&A session. Next slide, please. We are joined today by our presenters, Liezl Foss, Dr. Bridget Freisthler, Dr. Liz D'Amico, and Dr. Luana Colloca. Liezl Foss is a HEAL steward in the data management group at RENCI. Foss has 5 years of experience working in both industry-led and academia-focused clinical research trials. Working previously for a large contract research organization, she was involved in the management of several study sites within a given trial, ensuring that proper documentation and clinical procedures were followed. She has worked closely with PIs to ensure that study visits and other study-related tasks run smoothly and efficiently during her time as a clinical research coordinator for the medical university. As a HEAL steward, Foss integrates her background to support investigators in HEAL ecosystem by providing guidance and strategy for data management. Dr. Freisthler is a professor in the College of Social Work at the Ohio State University and principal investigator of the Healing Community Studies in Ohio. Dr. Freisthler's research focuses on how situation, social, and location characteristics affect the use of child abuse and neglect. Specifically, she is interested in how microcontext, for example, where a person is during a particular event, shape behavior, such as parenting or substance abuse. These microcontexts might include where and with whom parents drink alcohol, use marijuana, or experience higher levels of stress. All of these may affect parenting behaviors. The goal of this research is to design, implement, and evaluate intervention strategies that reduce substance misuse and child abuse and neglect. Dr. Freisthler will speak about her experience sharing sensitive, private administrative data in a HEAL-funded study. Dr. Liz D'Amico is a senior behavioral scientist at the RAND Corporation and is a licensed clinical psychologist. Dr. D'Amico is nationally recognized for her work developing, implementing, and evaluating interventions for adolescents and young adults. She has grants focused on prevention for substance abuse for urban Native American adolescents and young adults that integrate motivational interviewing with traditional practices such as beading and Native American cooking. She is currently conducting a large clinical trial to address suicide prevention among Alaska Native young people in Alaska. Dr. D'Amico will speak about her experience sharing sensitive data in a HEAL-funded study working with tribal communities. Dr. D'Amico will also be joined by her colleague, Dr. Daniel Dickerson, who will be joining us for the Q&A session. Dr. Luana Colloca is a professor in pain and translational symptom science and director of the Placebo Beyond Opinions Center at the University of Maryland School of Nursing. Dr. Colloca has conducted pioneering, groundbreaking studies that have advanced scientific understanding of the psychoneurobiological bases of endogenous systems for pain modulation in humans. As a result, Dr. Colloca has developed an international reputation as a leading scientist for advancing knowledge of the neurobiological mechanisms of placebo and nocebo effects with a multifaceted approach, including psychopharmacological, neurobiological, and behavioral approaches. Her research has been published in top-ranked international journals, including Biological Psychiatry, Pain, JAMA, the New England Journal of Medicine, and Lancet Neurology. Dr. Colloca will speak about her experiences sharing sensitive genomic data in a HEAL-funded study. Please go to the next slide. The objectives for our webinar today are to provide an overview of sensitive data types and nuances related to the requirements for data sharing, to emphasize the power and importance of sharing metadata in data management, to present HEAL world scenarios and lightning talks, and to showcase various resources and tools. I will now pass the presentation over to Liezl.
Thanks, Sarah. Next slide, please. All right, let's take a deep dive into understanding sensitive data types and the intricacies involved in data sharing requirements. Next slide. So in our latest webinar on sensitive data, we gathered audience feedback to identify additional data types of interest. We learned that HEAL researchers requested more insights on private and administrative data, genomic data, data containing intellectual property, such as SBIR or STTR grants, data related to populations at risk for social and cultural stigma, such as, but not limited to, the American Indian and Alaska Native communities, substance use disorder patient records, mental health patient records, formerly incarcerated individuals, data on sexual orientation and gender identity, specifically members of the LGBTQIA+ communities. So it's crucial to recognize the specific NIH data sharing requirements relevant to these sensitive data types. To assist HEAL researchers, stewards have published, stewards are creating a resource page regarding the sensitive data decision-making process. But we also have a resource page regarding these sensitive data types, which can be found on our HEAL Stewards resource page. And we'll paste the link to that on our chat. Next slide, please. As a refresher, this is a screenshot of the summary of the NIH Data Management and Sharing Policy, which mandates all studies to submit a data management and sharing plan, also known as a DMSP, emphasizing the need for maximum feasible sharing of scientific data with allowances for justified exceptions. And so this presentation focuses on understanding these nuances in data sharing expectations. And as I mentioned in the last slide, to assist our investigators in navigating the nuances to these expectations, we've created a decision tree, which will be an interactive tool published later this month. It will walk users through the decision-making process of the data sharing expectation. Essentially, the decision tree will lead to 3 terminal outcomes. One being that the NIH data sharing requirement applies to you after proper de-identification and or compliance with the GDS policy. Another terminal outcome is the ability to share these data along with secondary data types is contingent upon specific contractual agreements such as DUAs and other applicable terms of use. And the last terminal outcome to this decision tree is acknowledging practical and legal constraints. The NIH has guidelines for data types where data sharing might be limited. And so despite these limitations, sharing metadata remains a valuable contribution to the HEAL data ecosystem. Next slide. To illustrate the power of sharing metadata within the HEAL data ecosystem, I'll provide a high-level overview of how your study's metadata is used within the HEAL data ecosystem. Again, as a reminder that despite data sharing limitations, sharing metadata remains a valuable contribution to the HEAL data ecosystem. Next slide. And as a quick refresher, the HEAL data ecosystem is a distributed model facilitating data sharing across all HEAL-funded studies via the HEAL platform and HEAL semantic search. Both of these tools leverage metadata to power the process. Remember, metadata is data about your data. So when registering your study with a HEAL platform, you'll complete a CEDAR form to provide study-level metadata. In addition to study-level metadata, there's variable-level metadata detailing your study's collected variables. And while submitting variable-level metadata isn't mandatory, it greatly enhances the capabilities of the HEAL semantic search. And at the end of this presentation, we will provide you with a link to guide you on registering your study and where to submit your variable-level metadata. So I hope this slide was able to highlight the significance of metadata in cases where actual data sharing is constrained. Next slide. And so I'll return our focus on sensitive data nuances. We'll now enter a segment featuring lightning talks by 3 HEAL investigators. They will each share their experiences with handling private and administrative data from American Indian and Alaska Native populations, and genomic data. A Q&A session will follow, so please use the Q&A chat box during the presentation and we'll address them at the end. Next slide. So without further ado, I'd like to invite Dr. Bridget Freisthler from The Ohio State University, who will be talking about her experience with handling private and administrative data. Take it away.
Thank you. Next slide, please. So I'm going to talk a lot about the process of developing data use agreements for the Healing Community Study. The goal of the overall study is to reduce opioid overdose deaths by 40%. Next slide, please. To give you a sense of the task, there are 4 research sites and a data coordinating center. There were 109 unique measures that we were looking to build across those sites. And we had to identify a whole bunch of different administrative data sources. In this particular study, most state agencies. The DUAs had to be negotiated separately with each agency because they had different requirements around data sharing that were important to their agency. Next slide, please. So rather than just go through all of these questions, I'm going to sort of give you some context and more information related to what we were able to do and how we were able to do it. But these are some of the questions that need to get answers. It is rare for state agencies to give unlimited open access to their data. They're often unwilling to share any kind of code book or document listing the data elements. That's especially true in Ohio. So before you go and start thinking about, go asking what data you need, do your homework. Have you seen these data used before? If so, reach out to those individuals and see what is their process. How do you have to go about getting these data? Who are the key people you might need to ask for? When you're doing it, think holistically about your study question. You want to ask for what's going to be both your numerator and denominator if both come from the data set. So if you're looking at rate of individuals with opioid use disorder who have prescriptions for medication for opioid use disorder, then that requires you to make sure you know who has OUD, the denominator, and also who has that prescription for it. Also think ahead of time, is this going to be a one-time data drop or will it be ongoing? Data systems change. In Ohio, we're having the experience that our our agency that handles Medicaid data has upgraded their system, and we have not been able to receive any updates since last December 2022. And those updates were lagged by even 6 months. So the last actual data we have is through the end of June 2022. Next slide, please. I have found personally that if I can get someone who works with data directly on a regular basis, I can learn more about what is and is not available or feasible, and whether those data elements have any limitations. There are some data elements that never get completed or are sporadically completed by the users. So if that is a primary variable of interest for you, it's really good to know ahead of time that you're probably going to have to think about something else because those data just aren't available. Many state agencies have data councils or governing boards that weigh in on requests for the data. If you have someone at the agency that supports your research, that is a champion of your research, they are often in the room when these discussions are being had, and they can advocate and answer any questions about the research you're trying to do. Some organizations will have their own IRB process. we've seen that at the state of Ohio as well. So you must complete their IRB process before you can receive approval for the data. And in some cases, this is before even completing a data use agreement. Next slide, please. When it comes to federal, state, and local restrictions, there are often different laws and statutes that you're trying to think about. Some of the ones that are universal have to do with how agencies interpret HIPAA or 42 CFR Part 2, which places restrictions on substance use disorder treatment. In Ohio, we've generally not been allowed to get access to treatment data. It's been very limited. But Ohio has set up their own sort of data ecosystem that's cloud-based. And because they can control access to the data, they are now letting us access some restricted data, 42 CFR Part 2 agency. But this will differ across agencies. We have some agencies that allow us to use the individual level data, and we have others that just do not. And so any of the measures we need, the study is analysis unit is county. So any data we need, they will aggregate it and share it with us, which makes it really difficult if you have to go change data definitions, because you have to go back and ask them to recreate those data sets with the changes in mind. Suppression is also something that's differed across our 4 sites. States who have access to raw data can often include all the data in the cases for analysis, but just can't show values for individual counties where the data has met the suppression criteria. In the case of the prescription drug monitoring program, in 2 states, there's a state law or statute that says values between 1 and 4 cannot even be shared. And in Ohio, that agency is one of the agencies that aggregates the data for us. So we don't even get data when there are only between 1 and 4 cases. Next slide, please. So thinking about what we've learned, data use agreements can take a really long time. I've had DUAs take over a year to complete. Depending on how the DUA is written, for example, if you include exactly the specific variables you want, any modifications will require an amendment. And so for some agencies, they only allow you to negotiate these changes on your DUA once during the year. If it's February and you realize you're missing some variables and you can't renegotiate until September, you have substantial time that cuts into any data cleaning, curation, or analysis. Sometimes you can get agency to agree to categories of variables. For example, sociodemographics, without specifying those exact elements. That way, if you need another variable, you won't need an amendment of the DUA to receive those data. The other thing that you want to think about, particularly for researchers where publishing is important, does the agency require approval of any manuscript sent for publication? In HCS, there's a range of 30 to 45 days that are specified by the different DUAs. Some sites will rerun some of the data to make sure the numbers that you're using in the manuscript are the numbers that they get. Just last week, I heard of a DUA being negotiated where the expectation is that if the agency has an alternative explanation for the findings, it has to be included regardless of what it is. So you really want to pay attention to what's going on in those DUAs. Data lags are also something that's really important. In HCS, the data lags are from 1 to 11 months across the consortium. If you only have a 3-year study, but you have an 11-month data lag, this could cost you really time for analyzing the data and for finishing the project on time. Next slide, please. Ultimately, one of the most important things is developing relationships and trust with the individuals who have access to the data. Overwhelmingly, most agencies don't want negative press. So they are worried that people who are using the data don't understand the intricacies of the data, and it's going to cause misinterpretation. So some ways to get started with building the relationship and developing trust. I'm interested in studying these questions. What would be helpful for your agency? Be realistic what you need. Share with them what you've learned. And then in sum, treat this as a partnership and not just a data grab. Thank you. And I'm going to turn it over to Dr. D'Amico.
Hi, thank you, Dr. Freisthler. My name is Dr. D'Amico. I'm a licensed clinical psychologist and I'm really honored to be able to speak with you about our project today. My co-PI is Dr. Dan Dickerson, who's an Alaska Native addiction psychiatrist at UCLA. And together we've worked for many years to address health and well-being among American Indian and Alaska Native youth, young adults, and adults. Next slide, please. Today, I'm honored to be talking to you about our Takuna study, which is Traditions and Connections for Urban Native Americans, that is funded as part of the HEAL Initiative. Next slide. Just to give you a snapshot of our project, this is a large randomized control trial that we just completed the baseline data for, and we have 541 American Indian, Alaska Native emerging adults who have participated, who live in urban cities across 37 states. As many of you know, when the pandemic hit, many of us had to pivot our projects. And so our in-person intervention became virtual, which actually ended up being a really wonderful thing because, as you can see, we were able to reach many people across the United States instead of in just one area. As part of the project, we have participants complete surveys over a 1-year period, and all of the participants in our study receive a cultural intervention. So half of the participants receive a 1-hour cultural health and wellness workshop, and the other half of the participants receive our Takuna workshops, which are 3 1-hour workshops focused on understanding how to get support from your social networks, and also focusing on different traditional practices, including discussion of cultural identity, Native American cooking, and prayer. And we also have virtual wellness circles, which are put on monthly by our elders that address a variety of health and well-being and traditions, including storytelling, hoop dancing, Native American plants, and it's another way for participants to learn more about their culture. Next slide, please. Our main aims for this project are to understand outcomes across these 2 groups. As part of the HEAL Initiative, our primary outcomes are focused on initiation and escalation of opioid use. We also are looking at other alcohol and drug use, including cannabis and alcohol, looking at the time people might spend around people who use. And we have some important secondary outcomes that focus on functional well-being, including spirituality and cultural connectedness. Next slide, please. I'm sure, as many of you know, there's a long history of inappropriate use of data with American Indian and Alaska Native samples, and it's not something that happened long ago. It's still happening recently within the last 40 years. There's been misinterpretation of data, using data to stigmatize populations, and this has caused great harm to communities. So, as Dr. Freisthler also mentioned, it's critical to involve the community before the funding even begins to ensure that there's appropriate data reporting and sharing. Next slide, please. One of the things I want to point you to is this wonderful article resource at the bottom here by Liezl and Soto that actually really discusses the issues of thinking through data sharing with urban American Indian and Alaska Native people. And it's a wonderful resource that addresses some of the issues I'm going to briefly be talking about today. But one of the issues is data sovereignty. So briefly, sovereign status is how decisions need to be made after consultation with federally recognized tribes. So for example, as part of our cooperative prevention agreement, there's another project run with Dr. Kelly Comroe and the Cherokee Nation. And Cherokee Nation is intricately involved with their IRB in making decisions on all of the procedures and protocols for that project. Now, it's a little bit trickier with urban areas because sovereign status is only recognized on tribal lands. And so we have to think very carefully about how to approach data sharing in ethical ways so that we can represent people in appropriate ways in urban areas. Next slide, please. So our Takuna project has been really proactive about this and thoughtful and really focused on ensuring that the community is represented. We work closely with our community partner, Sacred Path Indigenous Wellness Center, which is headed up by Dr. Carrie Johnson, who's Wahpeton, Dakota. And she's worked with Dr. Dickerson and I for many years on numerous projects to ensure appropriate recruitment, retention, and data collection of urban American Indian and Alaska Native people. We also have an elder advisory board that we meet with monthly, even before the project began, to discuss these important issues. Out of conversations with Sacred Path, our board, and the community, we realized that not only is it important to have a clear understanding of the implications of data sharing, but participants also need to have an option to opt out of data sharing on the consent form where they can still take part in our project, but they don't have to share their data. So out of the 541 participants that we have in our study, only 25 of them opted out of data sharing on the consent form. And to our project, this really highlights how important it is to explain clearly the implications of data sharing, and that when it's explained clearly, most people feel safe and comfortable doing this. We also focus on other approaches to ensure that the community is aware of how the data are being used. So we discuss our research findings with communities. Every publication in a peer-reviewed journal is put together in a one-page lay document and disseminated to the community with our findings. And many of our elders also are co-authors on our publications. In addition, Dr. Dickerson is part of the HEAL Dissemination Access and Research Work Group that is focused on data sharing among all the projects, and particularly trying to think through procedures for data sharing for American Indian and Alaska Native people. Next slide, please. So as I mentioned, the most important thing is to be proactive and think through with the community ways to share the data that feel comfortable, appropriate, and safe. And one of the ways that our project has done this is to create an urban intertribal Native American review board that helps protect tribal sovereignty and helps think through the appropriate use of data with our urban American Indian and Alaska Native participants. We have a lot of work still to do, and we're in further discussions with the National Institute of Health and tribal communities to really think through how to establish formal guidelines for research that's conducted among American Indian and Alaska Native people in these urban areas. So thank you for being here today to learn about this, and Dr. Dickerson and I look forward to addressing questions at the end of the presentation. And now I'd like to introduce Dr. Colloca.
Thank you very much, Liz. It's a great honor to be today here together to talk about HEAL secondary data analysis and sensitive analysis. I plan to talk about our projects and multi-PI with Susan Dorsey, and we both have been working on genomic data. This project is a parent R21 with data collected from an R01. Our goal is to understand the predictors of placebo effects. Next, please. So what placebo effects are? So we use placebo effects as a proxy of endogenous pain modulation. When we receive any treatment, not merely a placebo, a set of mechanisms are activated in our brain, in our body, to modulate pain. And the power of the APM is that we can reduce chronic pain, acute pain, and in particular, we can understand descending pathways to modulate pain. Recent studies have investigated genomic and pharmacological effects in response to placebo. Our goal here was to identify gene expression patterns associated with placebo effects. So for doing that, we had to ask our participants that were in-depth phenotype for clinical, sociodemographic, biological characteristic to also collect blood that eventually was saved for a future grant, additional resource to analyze later on as secondary data. Next, please. So first we had to work with our participant, but before starting the project with the local IRB as well as the HEAL initiative in terms of sharing data and obtain the right informed consent so that participants would feel comfortable to have this blood drawn for future studies. So to optimize the data and the safety monitoring plan, we use REDCap dataset that is HIPAA compliant. And also the PI had a direct and total control of the system while we were acquiring both the ethical aspects and the data. In terms of IRB consideration, next please. We were sitting down with our patient and we explained them the relevance of collecting these samples for future studies. So we were telling them in the consent, this is the language we use when a researcher shares information with other researcher, is to assist more powerful research studies. So we explained that, for example, for a GVAS study, we need 1,000 of participants, but also we were collecting blood for RNA extraction, where also smaller data set can be informative in terms of scientific goals. So we told them that we were creating this database that eventually was shared through the database of genotype and phenotypes with NIH. And so they were aware of this possibility. The NIH Data Access Committee had approved that, and the researchers were also approved to collect this kind of information while preserving sensitive data like the social security number from being shared through this scientific data set. Next. Importantly, as we mentioned before, we also allow the participants to choose. You may choose to not have your saliva, and the same for blood, sample store. And if you choose to not have the sample store, you can still, be in the study. And also we give them the opportunity to complete the study and destroy the saliva and blood. The saliva was used to extract DNA and blood to extract RNA. And so they were able to initials the option that made them more comfortable. I consent to indefinitely store, or I do not consent. And we explained to them in verbal and written form that in order to conduct this analysis was important for us and other university, government, drug, health related company to have access to this data. We explain how careful we are in preserving privacy, confidentiality, but I think the most important aspect is truly to give them the option to destroy the samples or do not be stored indefinitely for future studies. Fortunately, over 802 participants, only a few decide do not consent to storage. And so we were able to complete the study and also have exploratory samples for this R21. Next. And with that, I would like to show some of the data from the initial R01 and the R21. So this show our data set. Behaviorally, we assess placebo analgesia in over 400 health controls and temporomandibular pain disorder patient. The goal was to understand when we study, real pain, people who suffer from chronic pain, do the phenotype of placebo analgesia match physiological conditions where pain is experimentally created in a laboratory setting. So up to that point where we discover many aspects of placebo analgesia, from ethnicity difference, sex difference, the impact of chronic overlapping pain conditions, we were able to focus on a smaller data set for the R21 and create two cohorts, a discovery cohort and a validation cohort, to perform this RNA study. Next, please. So these are all the steps that we perform in order to extract from this secondary data analysis. From the blood, we extract RNA, and the RNA sequencing allows us to identify over 10,000 genes associated with proteins. From these 10,000, about 600 were significantly associated with placebo effects. But the goal was to validate that, and you can see the number here in a validation cohort. So when we run the validation cohort, up to 6 genes that are described and presented here show a significant association with placebo effects. Also, we run what is called unbiased enrichment analysis to discover how these over 600 genes associated with placebo effects are enriched in pathways that are meaningful. Next. And this is a graphical representation. We found that pathway associated with the regulation of split and robo, Huntington disease, ribosome biogenesis, cellular response to chemical stress, mitochondrial translation inhibition are the top pathways that become relevant when naturally people can have access to the sort of inner pharmacy that allow the endogenous modulation of pain. And as I mentioned, the proxy is being a placebo responder in a procedure run in a lab, in our lab. So this pathway become relevant to determine those who respond to placebo effects. Next, please. So the next one is AIM2. So we will share all these results through the big apps, but also our intent is to make this data available to other PI, so that eventually we can have GWAS data along with single nucleotide polymorphism results, and larger cohort of participants can allow us to have a much in-depth phenotype of placebo as a phenomenon. And all these are the things that we plan to share with NIH. Next, please. And so in terms of acknowledgement, definitely we wish to acknowledge the National Institute of Dental Craniofacial Research for the first one, allow us to truly understand more about the placebo in a condition that is orofacial pain affecting young people, and also a disease that continue to be orphan. We don't know the etiology and there is no consensus on the treatment. Understanding how endogenous pain modulation can help mitigate this pain is definitely important. And also I present here some of the link to the R01 generated paper where you can read more about placebo effects related to TMD. And our goal is to disseminate this research to this scope. Recently, we made open access Oxford University Press book where we summarize the results of data sharing, the relevance of molecular mechanism associated with placebo effects. And with this, I would like to thank all of you for organizing this important webinar today. Thank you.
Thank you to all our lightning speakers. I will showcase the resource slide next. So next slide, please. I know there was a lot of information with a lot of links and a lot of resources that were shared. So foremost, make sure you visit our HEAL Stewards resource page, which is a key resource for managing sensitive data within the HEAL Initiative. It features essential guidelines and tools for secure data handling. And as I mentioned earlier in the presentation, we're excited to announce that a new tool will soon be available, which is the sensitive data sharing requirement decision tree. And so keep an eye out for that portal for its launch. Additionally, please check out the HEAL PI checklist. We'll paste the chat in the link to streamline your study's registration and metadata submission. And lastly, we'll send out today's presentation slides via email, providing you with an easy access to all the discussed links and resources. Next slide. And so the key takeaway, we were able to hear about various types of sensitive data with their own considerations and resources. We were able to hear about how our HEAL investigators can achieve harmonious balance between the goals of open science and the diligent management of sensitive data. And our HEAL stewards' contact info and form can be found on this slide. And so now I'll turn it over to Sarah, who will be facilitating the Q&A portion.
Thank you, Liezl. We'd like to invite all of our presenters to turn their cameras on for the Q&A, and we would also like to invite Dr. Daniel Dickerson to join us. He is a PI for the Ticuna project and works closely with Dr. D'Amico. Our first question is a general question that I will open up to all panelists. From one of our attendees, I'd like to better understand how people have shared qualitative data in the past. Are there recommended techniques for researchers to share qualitative data when there is a small sample size to ensure anonymity? Yes, Liezl, I guess if you want to take that first, and then we'll open it up to other panelists.
Sure. My answer will be coming from a data steward perspective. So yeah, small sample sizes often raise issues around the privacy and anonymity of participants, especially when the data is specific enough to potentially identify individuals. So we've had this discussion with the NIH and my fellow stewards. We advocate for sharing of summary or aggregate data, coupled with strict adherence to de-identification processes as outlined by the Qualitative Data Repository, also known as the QDR. It's important to note that the QDR's de-identification guidelines are different from the HIPAA's standards, which focuses specifically on PHI or protected health info. The QDR's guidance applies more broadly. It covers a wide spectrum of research data types beyond just health-related info. And in addition to these measures, stewards suggest considering tighter access control mechanisms, which includes requiring contracts. We've heard from our speakers today, like using data use agreements or really tailoring data use agreements for accessing specific data or utilizing repositories with secure enclaves, where data access is under controlled conditions. And so for detailed understanding of the QDR's de-identification requirements, we can paste the link to the QDR de-identification. And I can open up the floor to other speakers to see what they have to say about this topic.
Yes, I have some feedback on that. Yeah, thank you for the excellent question. I think with Native American populations, this is highly relevant since Native American communities are smaller and ensuring confidentiality is hugely important. So what we try to do is when we focus on our publications of qualitative based data, we of course don't mention names. We also very rarely, or if ever, mention one's tribal affiliation, because that may open up the opportunity, that may open up potential for confidentiality, stigma issues, and further stigmatization. We also try not to include names of communities or cities. So if we have a quote from someone, I'll just name a random state, Wisconsin, we'll say from a northern city in such state, we may refer to a community sample or individuals. We also really do our best to include and to utilize community-based participatory research principles, and including community members and Native researchers and staff who may have further insights into what's being reported in qualitative data. There may be some bits of information that could identify a family or individual based on what subjects being talked about. So we really try to vet and share and discuss all reported quotes as a team as well. So those are my thoughts on that really good question. Thank you.
Okay, thank you very much for your responses. We'll move on to our second question, which is, can the speakers talk about the specific pain points that they have experienced in navigating the data use or sharing agreement process at their institutions? Do you have suggestions for researchers in navigating this process or suggestions to improve the process? Dr. Freisthler, would you like to start with that one question?
Absolutely. Thank you, Sarah. So one of the things that I found consistently is really getting access to the attorneys. They have a lot on their plates and they don't always respond very quickly. So figuring out who is going to be the right attorney that's going to address your request is really important. And then having conversations and reminders saying, where are we with this? Where is it in the process? What do you need? I also suggest that whenever possible, if the agencies you're working with have a standard DUA and you have a standard DUA, that you grab those and look at them so that you can try to figure out where does the language differ and what are the possibilities that you can get them to either agree not to put anything, be silent on it in the DUA, or, try to negotiate something ahead of time so that you don't have the attorneys just talking with each other. Also, really pay attention to the things that are most important to you to make sure those get in DUAs. Oftentimes, it can be flexible about publishing and who owns the data and how you want to use the data. And so if you can get them to sort of listen to what you need, or you have a sense of what you're looking for, you can come in sort of with that and say, these are the 3 conditions that really need to be met. And you tell that to your attorney, and then they know, like, I might be able to negotiate over here, but I need to keep these core pieces really important. So those are just some things that I try to do when I'm starting the new DUA process.
I agree very much. It is very important to start the process in advance, because often the questions that the lawyer raise are beyond what we anticipate. They focus on details that we may not have even thought about. And also can be interesting to tell them our deadlines, that, okay, they continue to negotiating back and forth, but we have our timeline. We want to start the project. We want to do the analysis. So to show that somehow we are engaged and we are part of the process, the data use agreements can't be merely in the hands of the institutions. You guide them with describing your research in lay language so that they can be engaged in what you do. Start in advance and tell them we expect to start in 2 months, 3 months. We would like to have this finalized. We expect to generate 2 papers. The authorship will be shared as this, and that. So to be very upfront with expectations in terms of timeline, intellectual property, but also to show that you have already built trust with that institutions. It's not something that is coming out of the blue, because eventually you got a grant or you have already a paper that you are working and collaborating on and so on. So, but sometimes the length can't be anticipated and maybe longer than we thought. So definitely try to start in advance with the process.
Thank you for your responses to that question. Our next question, what are specific considerations when using data with American Indians and Alaskan Native communities? And how can we respect cultural sensitivities while promoting open science? Dr. D'Amico and Dr. Dickerson, I'll start with you.
Yes, I have a quick comment on that. I think one quick thought that comes to mind is that we really have to be very careful in how we describe Native American communities. Unfortunately, Native American people have experienced significant hardships and including historical trauma, high rates of violence, suicide, substance use, and other health conditions. And a lot of that's been reported over and over and over again. So we fully know that that's true. So what's really occurring now is more of a strength-based or solution-based way of how we go from here, how we report data, how we're generating data, and how we're using it to help, really try to improve various health conditions. So we're kind of looking more on the positive slant, if you will.
Well, and I think that's really important what Dr. Dickerson is bringing up, because I think for many historically marginalized populations, we see papers written, they have high rates of this and this, when really communities want to understand how to address challenges and how to increase resilience. And so we've really learned from our community partners and how to think through how to increase protective factors and how to, create programming that's focused on positives like cultural, spirituality, connectedness. And it just, I think, really helps communities and working with our elder advisory board, like Dan and I have, and in ensuring that the community feels represented and that it's an appropriate way of helping them think through the challenges that they might be experiencing.
Thank you for your responses. For our next question, what are some of the best practices to de-identify genomic data while preserving its research value? Will removing HIPAA identifiers be enough? Dr. Colloca, would you like to start?
Thank you. We study both health controls, and we match for race, sex, and age, but also patient. And patients come very often from our hospital or Brotman clinic for TMD, where their data become part of the clinical data set. Our practice was to remove the HIPAA identifier from our participant and creating a code, But also we decided to have two codes. All the patients that were phenotypes at the clinic within our hospital system receive a first screening code. And then when they came to the lab for all the phenotype related to research, we associate them with another code. That increased somehow, the privacy, because all the clinical data that they may have access, other clinicians may access as part of the AP or the chart that the hospital use, were dissociate from the research-based or the genomic data that eventually we share with NIH. So to me, creating two layers of de-identify participant can help, to ensure privacy and confidentiality, because being part of a patient data set at the hospital system is different than being part of a research system. So it can help to have two distinct codes. Thank you for the question.
Our next question, for Indigenous data, how useful do you find the CARE principles? And I will open this up to anyone on the panel.
Do you want to answer that, Dan?
Yes, yes. I think that's a very good question. I'm not as I haven't referred to the CURP principles in a while, but I know they are a really, really good resource. I think that's being used in many communities. I think they're fundamentally have and cover the right kind of topics that we're talking about today. And what's really great is that, you know, they're really trying to look at a global sense of our ways of approaching data, because we are part of a global community. We are part of a, you know, a really international based community. So the more that we're able to really look at these principles in a sense, in a way that recognizes all of our rights, indigenous rights, I think is all the better.
Well, and I think, you know, an important thing that we've been dealing with our projects with urban American Indian Alaska Native communities is really thinking about how people can have control of the data. And so that was why we created this proactive, this review board for our study, so that there are ways that people have control of the data and can assure appropriate use of the data in these situations where there's not something set in place. And so, you know, representing the community is most important. And I think these principles are able to do that.
Thank you. And we've included a link if you are interested in what the CARE principles are. Our last question will actually be discussing intellectual property. So I'm going to send this one to Liezl. The question is, can you go over the resources or tools that you recommend for researchers navigating the intersection of open science and intellectual property?
Thanks, Sarah. Yes. Foremost, I'd like to just reiterate that complying with the HEAL data sharing policy, or the NIH data sharing policy, which includes registering your study and providing associated metadata, doesn't impede securing intellectual property rights. Under the Bayh-Dole statute and the SBIR STTR program policy, inventors can retain their patent rights. And furthermore, these policies accommodate a delay in data sharing to protect intellectual property while simultaneously supporting research transparency. And therefore, sharing metadata aligns with HEAL's commitment to open science and doesn't conflict with your patent application process. Now, in terms of specific resources, we actually compiled a list of them that will be included in the email of the presentation slides. So that will include links to the HEAL Data Fair Sensitive Data Portal, as well as there's a deeper dive within the HEAL Data Sensitive Data Portal. There's a deeper dive on intellectual property, so you'll find the links to that. Another resource would be to look at the NIH DMSP policy, which offers plenty of guidance in managing IP in compliance with the NIH policies. And I think the last resource I could I'll include is a legal document, which the Legal Interoperability of Research Data Principles and Implementation Guidelines. And this document provides guidelines for legal interoperability in research data. And again, this will all be included in a slide or two when we send out the presentation slides via email after this webinar.
Thank you. I would like to thank all of our presenters, Liezl Foss, Dr. Freisthler, Dr. D'Amico, and Dr. Colloca, as well as Dr. Dickerson, for joining us today. We are very, very grateful for your insights and participation in today's webinar. In addition to our presenters and panelists, we would like to acknowledge the behind-the-scenes team that is listed on the next slide, who diligently worked to bring this webinar to production. Next slide, please. And we would like to thank the audience for your participation today in today's webinars. We hope that you are leaving with a better understanding of unique sensitive data types and how HEAL researchers consider and make decisions around sharing results associated with sensitive data. In addition to a recording of this webinar, we will also email everyone who has registered a list of resources. This slide deck will be shared, and a link to our Slack channel and Connect With Us form will also be located in the chat and in the email. We hope you will reach out to the HEAL Stewards team for questions or for support as you embark on your FAIR journey to maximize the value of HEAL data. We look forward to seeing you at our next webinar, which will be on Thursday, March 21st at 1 p.m. Eastern Time, and it will focus on research publication. Thank you again and have a wonderful day.